So, good morning, and thank you very much for coming. So, you might remember me from yesterday. My name is Johann. Look familiar. Yeah, so yesterday there was a quite formal presentation. Today will be a bit informal, so if you have any questions, just interrupt me. I would like to talk about Dynamin 2 and give you an overview on Dynamin 2, clinics, mutations, histology, and also talk again about the therapies, just to make sure. This was great yesterday, but just to make sure that everybody got the point, I would just reuse some of the very beautiful slides from Belinda. So, Dynamin 2. So, we have now in total approximately 125 families described worldwide with Dynamin 2 mutations. So, it's not as frequent as MTM1, where we have 500, 530 families that are described, BIN1, very rare, 15 families, and Dynamin 2, approximately 125. So this is, of course, only in the countries where molecular diagnosis is possible. So just to give you an example, so you're all from different countries. So in France, there are 60 million people living, and we have 2 new Dynamin 2 cases per year. Okay, so if you extrapolate this on the whole world, then of course, worldwide, there are many, many more than 125 families. But most of the families do not have access to a neurologist, to a molecular diagnosis. So this is clearly a disease that is underdiagnosed. There are hundreds and potentially thousands of families with Dynamin 2 mutations in the world. On the histology, they are quite easy to recognize if it is a Dynamin 2 case. So again, we talk about the biopsy. So we take the biopsy, then we do some chemical staining. This is what we call histology. And what we see very typically in patients with Dynamin 2 mutations is this here. So first of all, you see, of course, the centralized nuclei on this. So this is here muscle fiber. This is another one. And actually, this is a transverse section. So the fiber goes out of the screen like this. So this is here muscle fiber, okay? So that is a cut like this, section like this. So we see here that there is lots of centralized nuclei, but what we also see, and what you see on this picture, is that we have a very big fiber size variability. So here we have a very big fiber, there we have a very small one, big one, small one. Typical for Dynamin 2. Big fibers, small fibers, internalized nuclei. And on another staining, this blue staining, we have here typically what we call the spoke of wheels. So this is also very typical for Dynamin 2 cases. So based on the biopsy, we can already say, okay, this looks very much like Dynamin 2. Then we have to prove this, of course, so that we sequence, that we analyze the DNA. And what we see typically in Dynamin 2 cases is it is the same mutations coming up all the time. This is not the same like MTM1. In MTM1, 500 cases described, and all the mutations, so this misspelling on the DNA, they are evenly distributed over the gene. This is not the case for Dynamin 2. We have always the same mutations popping up, always the same mutations. This is what we call genetically a hotspot, a mutation hotspot. And this is here the different, so the 5 mutations that cover basically all the patients. So, for example, the most frequent one is this one here. So this is here the code of the mutation. This is what is your molecular diagnosis. So this means that at position 465, there is an R that is changed into a W. That's an arginine into a tryptophan. But this is not important. So this is the change. That is the position. This is what we need to give you prognosis, for example. So this is what you have on the sheet of paper when you get the molecular diagnosis. And this mutation here, this is found in 25% of all Dynamin 2 patients. It is quite a moderate central nucleomyopathy. When you have this mutation, it is not the most severe form of central nucleomyopathy. This one here is much more severe. This is at position 619, an S that is changed into an L, found in 10% of the Dynamin 2 patients. This is usually found in more severe cases. So what I want to say with these hotspots, is that as we have many families with the same mutations, we can give you a prognosis. If there is a new patient with Dynamin 2 mutation and we identify clearly the Dynamin 2 mutation, then we can give a prognosis. Because we have already 30 families in the world that are described with this one here. So if we have a new family with exactly the same mutation, then we can just look into our files and say, okay, what happened with the 30 families? What happened after 1 year, 2 years, 5 years, 10 years, 20 years? How was the disease development? And so we can give you a prognosis that depends, of course, on many other things, of the environment and all these things, but we can give you a quite good prognosis about the disease development in Dynamin 2. Now I would like to talk a bit about therapies. So this is just for the genetics and histology. So if you have any questions, then this would be the right moment to interrupt me. Otherwise, I would like to talk about therapies. No questions, so it must have been very clear, or you're very tired. Great, therapies. Because yesterday there was, for example, also the question whether the therapies would be mutation dependent. So if there is a therapy that is developed and you have that mutation, does the same therapy also apply for people with that mutation? And the answer is probably. From what we know in the lab, because what we did is that first of all we had to identify the genetic cause. So we found some 15 years ago dynamin 2. Then we tried to understand what is happening in the cell. That's the pathomechanism stuff. And from what we understand from the mutations is that they all have the same effect from what we know. But of course we have to study a bit more. So this would mean that a therapy that has been established for one mutation could be also suitable for the other mutations, because they seem to have the same effect. So to answer this question, whether the same therapy would apply for all. Okay, now let's come to the therapies, and let's come also to some basics, because some of you might ask yourself what's the thing with the centralized nuclei, why it's called central nuclear myopathy, why it's called myotubular myopathy. So I just want to come back to this point and then talk about therapies. So what happens during muscle development is that at the beginning of muscle development, we have some single muscle cells. So this is here some single muscle cells with the nuclei that are floating somewhere in the cell. And what very specifically happens in the muscle tissues and in no other tissue is that hundreds and thousands of these cells fuse together. And then they form a myotube. So this is here myotube. And in this myotube, we have lots of nuclei because all of these cells fused. And they are evenly spaced, and they are in the middle of the muscle fiber. And then there is a next step, it's the muscle maturation. And during the muscle maturation, the muscle fiber gets bigger, and these nuclei move to the periphery. We don't know exactly why, but they move to the periphery. Now, if I take a biopsy of somebody with such a muscle fiber, and we make a section, transverse section, we get this picture here. We have here these fibers, so 4 fibers, and the nuclei, they are in the periphery. If we make now a section on muscle fibers from a patient with central nuclear myopathy, we get that picture. That is different. So you see already that they are smaller fibers, and the nuclei are exactly in the middle of the muscle fiber. So it looks like as if we had made a section through myotube before the maturation of the muscle fiber. That's why it's called myotubular myopathy, because it looks like a myotube. But myotubular myopathy and central nuclear myopathy is the same. In the scientific world, we use only the term central nuclear myopathy. So it's the same disease, myotubular myopathy, central nuclear myopathy. But we use only central nuclear myopathy. Okay? But it covers also myotubular myopathy. And before coming to the important point for therapy, I would like to make a point also on the function of all these genes. So what is BIN1 doing? What is dynamin 2 doing? What is MTM1 doing? That is very important to understand therapy. And for this, we zoom in into a single muscle fiber. And MTM1 and BIN1 and dynamin 2, they are forming these T-tubules. So this is called a T-tubule here. So this is a very deep invagination that goes deep into the muscle fiber. This is necessary to transfer the signal into the muscle fiber for muscle contraction. So how does muscle contraction work? Your brain decides that you want to do muscle contraction. And this is an electrical signal. And so this electrical signal goes through your nerves to the muscle. But then it has to go deep into the muscle. And it can go deep into the muscle thanks to these T-tubules. They are formed by MTM1, dynamin 2, and BIN1. Okay, and now the signal reaches here the end of the T-tubules, and what is the signal doing? It's activating the ryanodine receptor. And the ryanodine receptor is a calcium channel. A channel is just something that goes on, and well, it opens and closes, that's a channel. So it activates the ryanodine receptor, the receptor opens, this channel opens, and this leads to the efflux of calcium. So calcium is kept in the cell in a little pocket, and when this ryanodine receptor is activated, all the calcium goes out of this pocket. And this is inducing muscle contraction. It's the calcium that is inducing muscle contraction. So how does muscle contraction now work technically? So there are some filaments, and when calcium binds to the filaments slide into each other. This is muscle contraction. So you see that muscle contraction depends on many different things, and it's a concerted work of MTM1, BIN1, and dynamin 2 that has to form these T-tubules of the ryanodine receptor that has to free and to liberate this calcium, and of the titin that is implicated in the filament sliding. So they all work together. They are forming a structure that is essential for muscle contraction, and the structure determines the function. And this is also the reason why people with mutations in dynamin 2 or MTM1 or CNM have similar diseases, because they all work together. Whether you have a mutation in one gene or in its partner, it will be at the end, it will be a similar disease. And that's why we have different forms of central nuclear myopathy, and they are due to mutations in genes that are collaborating. And that is extremely important to understand, because this also means one thing, that if you develop a therapy for one of the forms of central nuclear myopathy, you have good chances to treat also with the same therapy the other forms of central nuclear myopathy. Because they all work together. You tackle one, you might cure the other ones. They are all working in a network. So this is the point that I would like to make before now coming back to Belinda's slides. So as I told you, you remember the picture that I showed with the football field, so that we have the genetics, so we try to find genetic causes of the disease. We found them. We found that 20 years ago that MTM1 is implicated in central nuclear myopathy. 15 years ago, we found that dynamin 2 is implicated in central nuclear myopathy. And then we wanted to understand what is really happening in the cell. Why mutations in MTM1 or dynamin 2 are generating or causing central nuclear myopathy. So we were analyzing what happens in the cell, in the petri dish, but also in the animal models. And this is here Belinda's work here, that she found out that as all these genes are working together in the cell, well, there is also balance between these genes. And if one of the genes is not there anymore, then there is an imbalance. And this is what Belinda found out. So in patients with central nuclear myopathy due to MTM1 mutations, there is no myotubularin. And when there is no MTM1, tubularin, then there is an imbalance between dynamin 2 and MTM1. So that dynamin 2 seems to be too active, and this is obviously what is creating the disease. So the idea of the therapy was now, would it be possible to reduce dynamin 2 to treat central nuclear myopathy? Because if you have such an imbalance, you have basically two possibilities. You can either replace MTM1. This is the gene therapy that a dentist is providing. So they replace MTM1, or you can also tackle it in another way and say, can we somehow reduce dynamin 2 to rebalance MTM1 and dynamin 2? And so this was the idea of a therapy. And as Belinda explained yesterday, we had some mice without MTM1, and Belinda reduced then dynamin 2 in these mice, and she could rebalance then MTM1 and dynamin 2. And the mice had a normal lifespan and an almost normal force. So this showed already that this is possible, that you can rebalance either by adding MTM1 or by reducing dynamin 2. But if this works for MTM1 cases, this of course might work also for the dynamin 2 cases. Because what do we have in patients with dynamin 2 mutations? We have also an imbalance between MTM1 and dynamin 2. Although we have MTM1, but this dynamin 2 somehow is too active. But the same strategy would apply. We would then try to reduce somehow dynamin 2, to turn it down a little bit, to rebalance MTM1 and dynamin 2. So it would be here the same therapy for MTM1 and dynamin 2 cases. And all of these genes, as they are all linked, MTM1, titin, ryanodine receptor, so this might also apply for all forms of central nuclear myopathy, because they all work together. And then, of course, technically the question was, how is this possible? How can we reduce dynamin 2? And here, this is the same slides as Belinda showed yesterday. But I just wanted to repeat this because this is, I think, a very important point. If you want to reduce or turn down dynamine 2, you have two different possibilities. Either you do it at the DNA or at the protein level. The DNA basically is not doing a lot in the cell. It's just information. It's just a book. It's not because you have a manual for a great car that you have that great car. If you have a manual of the great car, it gives you the information of building a great car, if you have the capacities, if you have the material, if you have everything that you need. But it's just a book. That's the DNA. It's nothing else than just a book. It's just information. But what is really doing the work in the cell is what is encoded in this book. And this is the proteins. So this would correspond to this car. You might remember the picture. It was not the same car, right? But you might remember this greatest Australian car that was built from all times, so 25 miles per hour, something like this, I don't remember. So, and now we have two possibilities to reduce dynamine 2. So either you do this at the DNA level, so somehow you close a bit the book so that less cars are produced, so this would turn down the dynamine 2 production, or you would just tackle the protein by stopping the car. Okay, and the important point is, so just if I can summarize this, first of all, that for dynamine 2 we have these hotspots mutations, so we can give you quite precise prognosis on the development of the disease. And as all these genes are working together in the cell, when we develop a therapy for one of the forms of central nucleomyopathy, it can apply also for the other forms. So this is what I wanted to show you. And as this is a workshop, I'm very open to discussion now.